Our next speaker is Dr. Kris Bosse from Children's Hospital of Philadelphia, and I'm doing short because I forgot his intro. The topic for his discussion today is immunotherapeutic target discoveries. Dr. Bosse.
I thought she was kidding about that. Thanks, Patricia. It's a pleasure to. I think she's asked me every a few years in a row to come to Chicago. I've never been able to, in part because it's like she asked like a 2 days before the conference. So it's actually great that this coincides with the conference downstairs. So hopefully I can tell you a little bit about this topic, and I think it follows nicely with what Dr. Masner showed you. I didn't see it, but I can imagine what he told you about. And please do ask questions. It's not meant to be. You can interrupt me. You can ask me at the end. You can find me at the end. It's a little bit heavily biased towards what we do at CHOP and in my lab, but I'll try to give you a sort of view of how I see this subject. And being in Philadelphia and at CHOP in Penn, we're very familiar with the story. And this is this now young lady, Emily Whitehead, who you guys have probably heard about her and how she was really cured from CD19 CAR T-cell chimeric antigen receptor T-cell therapy. And this has been a very famous story, and she's actually at Penn as a college student now. And so this has really sparked interest in this area and using the immune system to fight cancers. And it's funny because this CD19 CAR T-cell is Kymriah. There's other types of this, and you'll see this all around. But really, neuroblastomas were the first really bona fide immunotherapy, and this is, you've heard of this, I'm sure, dinutuximab, and this is the seminal sort of paper. Don't worry about the specifics, but it showed that it was better than the standard of care at the time. But that was back in 2010, and so really, I think neuroblastoma was the forechild of immunotherapy per se, using the immune system. This is an antibody, and oops. That's really fast. And I'm sure Dr. Masner showed you about GD2 and CAR T cells, and this is just a paper that people have also tried to make these so-called CAR T cells against this sugar on neuroblastoma GD2. And don't worry about the details, but a lot of them haven't been very successful. Although there's some success here. And this is really a touchy thing. This is, I actually heard a dad asking Dr. Masner about this study, and this is a report in the New England Journal a few years ago. There was really the first time in neuroblastoma that people have showed that CAR T cells were effective. And if you look on the left, the orange is basically patients who've had complete response of their disease, and there's lots of them there. There's I think 40%. And on the right, there's, although looks maybe a bit subtle, but there's surely shrinkage of these larger tumors. And again, this is not something we had seen with CAR T cells in neuroblastoma before. So this was really exciting, and really I think the first proof that this could work in neuroblastoma or other solid tumors. And this sugar GD2, it's on most neuroblastomas. Again, you guys have all heard about this, I think probably because of dinutuximab. It's used very heavily in our regimens here, or other types of GD2 antibodies. But the truth is, this antibody's been around, or the knowledge of this being on cancer cells and antibodies in the blood of patients with cancer have been around for 40 years plus. And this, it's interesting what, I take this from the paper, this GD2, people worried about it being, on brain cells, or I think you guys are probably familiar with it being on nerve cells, and children needing pain medicines as they get GD2 therapy. So this was known way back when, and it gets at what I think about a lot, and what our group thinks about, is really finding these targets on cancer cells that could be targeted with different immunotherapies, and moving beyond something that's been known for a very, very long time. And it's not that it's not a good target, it's just that there must be other things, right? So that's sort of what my research does, and what I think has been really exciting about this area in the last decade or so, is finding new targets that really provide safety, just on tumors and not on normal tissue. So you can imagine the scenario where any of these little shapes can be a target. And what the idea is you see it on the left, but you don't see it on any of the normal tissue. So you can selectively get, whether it's a potent drug or these potent T cells selectively to the tumor. It's not like chemotherapy, right, where sure it acts on the tumor, but it also acts on your hair cells and in your mouth and in your GI system and all the other things that the side effects from chemotherapy. So this is a very targeted therapy. And I was lucky, and this is just, and you guys may recognize some of these names here, but about a decade ago, there was a huge effort to do just this, really find new targets for cancer immunotherapy, and specifically children. And this is some of the groups across the North America that were involved in this. And I was lucky to be at CHOP and John Maris's group. And it really came out of the idea that, I think you see a lot of television commercials and you hear about immunotherapies and they say, for small cell lung cancer or bladder cancer, and that's a different type of immunotherapy, and it really comes out that, and you guys know this, right? Adult cancers are very different than kids' cancers, and we live in a world where the cancers, the children, you guys know, your children and children we treat are, if developmental in origin, they contain things that are in developing tissues that normally get lost, but these tissues come out of aberrant, usually aberrant development, and neuroblastoma is surely that. So it's possible that there's unique things on tumors that aren't found in the rest of the more mature tissues in a child, and that's what we really tried to want to capitalize on. And I think. There's no doubt that there's been a lot of success in pediatric cancer therapy, but I think this group knows this for sure, that a relapse or some of the more high-risk cancers, there's still a lot of work to be done, and these are just some typical unfortunate curves that there's a patient with a relapsed neuroblastoma. We have to find better therapies, and so our hope is that finding new targets will really help us revolutionize cancer immunotherapy. And so. Don't worry about all these words, but these are different molecules on cancer cells, not just neuroblastoma cells. I've underlined some of the ones on neuroblastoma cells. And you guys you'll see these, GD2 again, you just heard about, but there's other trials where people are trying to target these different molecules with immunotherapies. And what the team did was really hopefully expand that to these different red ones. And it just helped really expand the targetable things on cancer cells. And I want to bring you through two different stories, one longer than the other, of these two molecules we got interested in. And these are neuroblastoma specific, although on other tumors. And, so as I mentioned, neuroblastomas, they come from developing nervous system tissues. So what we think is that some of the things that are on developing tissues normally get lost in a mature neuron, let's say, but persist on neuroblastomas, and this is a complicated depiction of that, but it's really how we think about this disease. And so this is sort of what we try to do. So this is a schematic, but you can look on the left, and what you really want, blue is high levels and white is low levels, and you want to find those things that fit in that top box. You want them to be high in the tumors and not on normal tissue. You don't want to give a CAR T cell or an antibody or that's going to hit normal tissue. You want to selectively go to tumors. And you want the things to be on the outside of the cell so that it can be recognized. And then if you find good targets, per se, you can make any of these different types of immunotherapies, whether it's a CAR T cell or an antibody, denatuximab is an antibody, or other different types of antibodies where you stick a drug to it and you can get drug potently to cells. So this is really the hope of cancer immunotherapy. And so we did this, and again, don't worry so much about the stuff on the left, but out of this sort of screen, we found this molecule GPC2, and you can see on the right this is just RNA, so what the main sort of building blocks of things in the cell. It's high in the tumor, and all the normal tissue, or most of the normal tissues, have very little. So that's sort of what we want to do, and the hope is, again, that you can build things to use this high level in the tumor to get things specifically to it. I'll skip that. And so if you look at all the different tissues in the body and brain, cerebellum, heart, you guys know what these all are. You can see the things to the left of the line are before birth, and birth is the line, and then after birth is a growing child. And so you can see how this GPC2 is in those developing cells, but not post-birth. So this makes sense to us. But then for some reason it persists in neuroblastoma cells. So, and this is a type of molecule where if you have it in the cell, cells grow faster. So we think it's something where we don't think the tumors can easily get rid of it. If you get rid of it with various tools we have in the lab, the cells don't want to grow. So that we think that's something a tumor's not going to be able to just shed and get rid of and evade the therapy. And someone's asking, what happens if you give all this dinutuximab? Will my child's tumor still have GD2, right? And I don't know the answer to, it's a complicated question, but the hope is if the tumor needs it, they won't be able to get rid of it without killing itself. And, you can do all the things in the world and find these great targets, but what you need is called antibodies. Antibodies are things we have in our blood. They fight infections, but they also bind cancer-specific targets very well. And you can do this in a lab where you can do these large screens where you find specific antibodies to bind cancer-specific targets. And this is the real data that shows that. Again, don't worry about the specifics, but we're lucky to work with groups within this larger team that allows us to do this. And from that antibody, you can build any of these various immunotherapies, a CAR T-cell, an antibody. Wrong button. And so one of the first things we've done with this is build this thing called an antibody-drug conjugate. And in the adult world, there's a lot about this, and I think we'll see more and more. And this is something I think is useful for you guys to hear, because I do think there'll be some trials where these are tested in kids and neuroblastoma specifically. And so an antibody-drug conjugate is this antibody. It's a protein. It's very specific for a certain thing. In this case, specific for this molecule, GPC2. And you can stick a drug to it. It can be any number of drug, and there's chemistry involved, but and you can do it like that. You can put 2 drug molecules or 3 or 4, and again, there's some science behind this. And then you can take that and you can imagine, if this antibody targets this thing that's specifically on cancer cells, but not on normal tissues, right? You can get this specifically to it. And if you imagine a kid, that has a tumor, you can get it specifically to the cancer, and it leaves all the other normal tissues alone. So you can imagine this is a very potent way to get a very potent drug. These are like chemotherapies, specifically to the tumor and not to the rest of the body. And so this is what it actually looks like in real life. This drug is very potent. It's like it's like certain chemotherapies you guys are familiar with, but not safe to give alone, but safe to give when you attach it to the specific antibody. And you can get things like this, where this is a mouse study, but tumors without drug, they grow very fast, and that's the black and the gray line. And then when you give this ADC, antibody-drug conjugate, you totally eradicate the tumors. They don't come back. So these things are very potent. These mice tolerate this very well. Oh, I have a picture. So that's sort of the sizes you can imagine. And you can do this across different models. And this is actually a small cell lung cancer. And you say, why is a pediatric oncologist talking to me about a small cell lung cancer? I think it's been important as we develop these things, to show other tumors that it's important with. And if you're a pharmaceutical industry like the ones I mentioned on the bottom there, interested, and we want them to be interested in neuroblastoma, of course, and I think we would love your help reminding them how important of a disease this is, because I think it's often hard to get industry partners interested in pediatric cancer. So we thought about this and really tried to prove how good this drug could be across different types of cancers, and I've gotten some interest. But I want to talk about CAR T-cells, because that's what everyone seems to know about nowadays, and you guys are seeing it in trials. And I can tell you about a trial I had an opportunity to talk about this morning downstairs. I won't tell you too much about the data, but you'll hear about this. And unfortunately, if you guys are faced with relapse disease in your child, that this will come up, I think, more and more about trials that are available across the U.S. So this is, on the left is a schema, and this is sort of how this is all done. So patients, kids, adults get their white blood cells collected through this leukapheresis procedure. It's usually it takes a few hours, but it's a whole day-long procedure. And that blood is then collected, and you can basically use, you do this in the lab, you can make patients' own T cells into these so-called chimeric antigen T cells, and then you grow them up to a lot of high numbers, and then you can freeze them back, and then when ready, you give them back to the patient. And you really turn these patient-specific T cells into tumor-targeting T cells that can get to the tumor, and then once they interact with the tumor, they secrete things that kill the tumor cells. There's different versions of these things on the right. Don't worry too much about that. And this is, again, what they sort of look like. But you can imagine on the right there, that's the CAR T cell interacting with the tumor. And through that interaction, the physical interaction, they releases the patient T cell gets activated. And T cells like to kill things. They kill viruses, but here they actually will then secrete things and kill tumor cells. Specifically, again, this is only happening when you interact with that specific molecule on the tumor cell. Let's see if this works. All right, so these are neuroblastoma cells. You're seeing an hour, every hour a picture gets taken. And it's actually interesting how they dance around a little bit. You never think they move so much, but they do. And so you'll see a bunch of dots get in here, right there. So those are T cells. And so the blue you're going to start to see are signs of cell death. So you see within hours. And you can see the T cells actually seeking out the tumor cells. And this is signs of the tumor cell dying. The blue is a molecule that gets released when the tumor cells die. And this is about a day after it added the T cells. And what you see is there's no tumor cells left. These are all just T cells now dancing around. And you see the blue goes away because all those cells are dead. So if you take neuroblastoma cells, put the T cells on in a dish, you basically kill all the cells within an hour. So again, it's not that simple, obviously, in a child or a model, but you can see how quickly and then these are just T cells dancing around at this point. But I think it shows you how powerful the drugs don't work this quickly. And again, this is an animal model, but you can get a sense. You give one dose of these CAR T cells, and your tumors go away forever. And again, it's obviously not this easy in patients, but the hope is that, again, we can get to this level of efficacy. So this is what I had a chance to talk about this morning. We're excited about this target. There's also GD2 CAR T cells trials, and this is our phase 1 study we have open at CHOP. I'm not going to talk more about this, but really trying to understand if this is a targetable molecule in patients with this disease and other diseases. And so this is, what we try to do is think about if we find these good molecules we think are safe to target, thinking about what other tumors they may be on. And again, I'm a pediatrician, so we focus on the things on the left. There's several brain tumors. Neuroblastoma is obviously the focus of this, some muscle tumors. Retinoblastoma is an eye tumor. And then there's some adult tumors on the right that we think this may be helpful for. But really trying to say, all right, if we got a good drug, where else can we extend this to? And this is what we spend a lot of time looking at. These are just a whole bunch of different tumors, and each dot is a tumor, and the higher it is, the higher this GPC2, this target is in it. And these type of data, you can see this helped us think about this is a really unfortunate, very aggressive muscle tumor. It happens in kids as rhabdomyosarcoma. So it gave us a sense, all right, well, maybe this is something we should think about. And I wish it was all this easy, but this is showing you some of the challenges of this type of thing. Again, getting at that question that Dad asked at the back of the room. So when you do this, sometimes you have tumors in the blue are the GPC2 CAR-treated tumors on the left. And you can see how they go away and then they start to come back. And then if you take that, this is in a mouse, you take the tumor out and the brown staining on the bottom left is how much GPC2 there is. So in the tumors that don't get the drug, there's a lot of brown, there's a lot of this expression of GPC2, but you can see on the bottom of that, those are the tumors that came back. They lose this molecule, even though it's important for the tumor. And so it become really important to not only do target one thing on the cell, you have to target multiple things, right? So if you target multiple things, can you kill those resistant cells that are driving this resistance and have lost your target? So this, I think, is where we're going to bring these trials, and I think will be much more efficacious. And the other thing, I think, getting back to my first slide, again, Emily Whitehead is this very exciting, just great story. But in leukemias, you have this world where your cancer cell is floating around in your blood, and it's terrible, but it's there. It's not embedded in a tumor. There's not a tumor mass. There's no immune cells. It's in the blood, and you give a CAR T cell via an IV infusion, and they're there, and they're able to interact. And it's sort of a best case scenario. So what we know with solid tumors is this is sort of the picture I think of, and there's a lot of forces within the tumor, and you guys know this for sure, right? These tumors are complicated ecosystems, and unfortunately, a lot of that is immune cells that don't want these therapies there, and they don't want these GPC2 CAR T cells or GD2 CAR T cells there, so they have forces to either kick them out, suppress them, and so you can imagine that there's all these extra hurdles to really get these therapies to the tumor, deep into the tumor, and allow them to persist in the tumor. Again, it's very different than leukemias. And so there's a way, I don't know if anyone heard Drew Weissman, who was downstairs this morning. He's most famous now for developing the COVID vaccine and the different RNA modifications that were in the COVID vaccine. And this is some work we do with him, and this whole idea is you can actually provide target GPC2 or whatever the target for the CAR T cells to outside of the tumor, and you can use some of his technology and stimulate the T cell or outside of this very complicated. So I think we're really excited about doing this and making it more like a leukemia, not so much like this complicated ecosystem in the middle. And so I'll just talk very briefly about another target, because I don't want to just be all focused on one. And again, I'm not, and this is a very biased view. But I think it's this is a postdoc in my lab, Patrick Shurk, and he's going to give this talk actually, or a better version of this talk downstairs on Wednesday. But we asked the question, well, if we can, if we had this whole list of genes, and that's the second to bottom thing, and we sort of ignored a lot of those, but he got really interested in this one in the circle there, and this is sort of what it looks like when you look at the same, neuroblastoma on the left and then all the normal tissue. So it looks very specific for cancer cells. And then what he found out, it gets complicated. This CHRNA, this protein is a complex of proteins. So it sits on neuroblastoma cells, but it's a sort of multi-subunit thing, and I'm trying to depict that on the right. And again, the specifics don't matter, but it has partners, and the partners change depending on whether it's a normal tissue or a neuroblastoma, and so. But all the partners are very high in neuroblastomas and not. So that may be a good thing, that you have a bunch of may give you more safety if you need multiple of these things present in the tumor. And oops, I keep hitting the wrong button. And you can imagine sort of on the bottom right, this and it's like you're looking down at this, and there's multiple A3, B4, all different sort of partners, and they do their channels and membranes. And what he did was build a CAR T cell towards two of these at the interface of these. So you can imagine a cell has to have both of them for this to work. And so even if a normal tissue brain cell, whatever it may be, has one of these, it won't matter. Like it won't bind to it. So, chemo, you have to come up with fancy names. We're not sure this is right, but we want to get at the point that you need the whole complex. And he can do this, and again, I won't show you too much of the data, but you can build cells in the lab with one or both of these two different complexes, and you kind of see it's like the video I just showed you, and here the cells that grow go up, and the cells that die go down. And so the only way this works is you have both of the things in the cell. So we're excited about this as being a different target for these cancers. And again, if you can combine these things, or I think that's what's going to be needed to really get to the efficacy of this, the Emily Whitehead efficacy. And so I'll stop there. I don't have to read this, but hopefully, again, I'm very biased as what we do in the lab, but I think there's a lot of good immunotherapy targets, and I listed them there, and you heard about GD2. There's other trials out there you may hear about. There's a new trial at CHOP that you'll hear about. It's a FOX2B trial run by John Maris, and it's similar, but it's targeting something that's taken from inside the cell and put on the cell by these molecules called MHC, which is part of a normal immune response. So it's a very different thing, and hopefully it works well. But you'll hear about these as because these trials are going to come to various places. I think one of the hard things is, and I got this question this morning, is how do we compare? One center has one CAR T cell trial, and another center has another CAR T cell trial. And I don't know, no one tells me what's better, which one should we do. And I think it's really hard for us because these are all, although they sound like they should all be the same, they're all very different. The T cells are made differently. They're made differently. How they're made, what they're made of is different, and it's really hard to compare. The patient populations they're used to treat are different. So I think it's a really hard thing to compare, and I think which is best. I don't have an answer for you. I think we're still learning about building off that GD2 CAR paper from a few years ago. That was really one of the first times where we saw that these could work in kids with neuroblastoma or other solid tumors. So I think we have a lot to learn. And I'll stop there and just put up my acknowledgments. It's we have a good team at CHOP, and we do a lot of this target discovery thing, and we're thankful to have really the cell therapy, the immunologists, to do this. So I'll stop there, and thanks for your attention. I'm happy to take questions.
So, Kris, I have a compliment and a question. First off, you're too modest. So you discovered a brand new protein on the cell surface of neuroblastoma cells, and you turned it into a brand new CAR T cell therapy that's not targeting GD2, but something else. And as Joe Biden once said, that's a big effing deal. So I just want to highlight that.
Gav is my biggest fan.
And then the question is, you talked a little bit this morning about some of the patients who've enrolled in the study, and I won't ask you to go through that, but just in general, can you talk about these kids and where they are in their treatment journey when they reach your trial? Yeah. And I think what I'm getting at as a bereaved father is you're offering great research and a great clinical trial, but you're also offering hope to kids who have been through so much over so many years, right? So can you talk about the patient population who are eligible for this study?
Yeah. So the study's open to any patient with relapse refractory neuroblastoma for now. We'll expand it to other relapse refractory diseases in the future. And we define that by sort of, patients who've failed standard therapy, and you can define that loosely, I think. But I think it likely means patients who, or how we interpret it, is patients who've gotten chemoimmunotherapy, so dinutuximab plus chemotherapy, and usually MIBG therapy. And then we typically have then would consider a patient that at that point patients have progressed through our standard relapse therapies. I think And I showed this slide this morning. The patients have all been much older. I think the median age is 14 years. So the patients who have had neuroblastoma for many, many years, I think the average of 33 cycles of previous therapy, and all the patients we've treated got this chemoimmunotherapy and MIBG therapy. So, that's part of a phase 1 trial, right? We don't know if there's benefit. We hope there's benefit. We think there's data in the laboratory that there's hope for benefit, of course. But the real goal is to make sure this is safe. And then now that we've gone through that phase, obviously we're very interested now in moving this closer to patients who haven't developed such resistant disease. And I do think there's a role, as we learn more about these things, is moving them. That's what's happening to dinutuximab, right? It's moving up in therapy and really getting these two patients sooner when their disease can respond to it better. I think, unfortunately, neuroblastoma, like other cancers, it's a relentless at times, and it can evolve to a point where a lot of things won't work, including T cells. So I think we'll get there. For GPC2, I think that our goal really is now to try to get this earlier and get this to a point where there'll be more hope for response and a less refractory tumor, unfortunately. Did that answer your question?
Any other questions? Go ahead.
I think on one of your slides, like you said that there was eventually like next generation GPC2 cars in the works. Like, what kind of timeline is that on?
Yeah.
And then two-part question, sorry. If and when, at what point would patients also be tested with their GPC2 expression to know if they're a good candidate for a GPC2 therapy or CAR?
Yeah, no, it's a good question. So I'll answer the second question first. We're doing that. So again, it's a lot of things are in retrospect. So we didn't. Well, right. So you want to get these into the clinic because we think they're going to be helpful. And I think many people in this room know it's not a trivial thing to do a biopsy, and we're lucky actually at CHOP we have great interventional radiologists who can do, I think, very safe biopsies in very tricky sites sometimes. But it's a hard thing to mandate. And then so if you say, all right, we need to do a biopsy to prove there's GPC2 or GD2 or whatever there to get on the trial, I think it's going to slow things down. But so we do it retrospectively now and look and try to understand if a patient responded or didn't, was there GPC2 or not. But again, sometimes these are historical biopsies from diagnosis 10 years earlier. So we have the tools to do it. This GD2, the thing that Dr. Masner talked about, is more tricky because it's a sugar and it's not easy to do the normal things we do to these things, but GPC2 can do it. So I think we'll get more information, but I do think we'll have to know to really get the most benefit, because if GPC2 is not there, it's not going to work. And even if it is there, it might still not work, unfortunately, because there's these crazy things happening in tumors, but it has to be there. So I totally agree with you. And then your second question is next generation therapy. So, I mean, the truth be told, that it's all my lab does right now is make second generation GPC2 CAR T cell therapies, and trying to understand why things didn't work or why things didn't work, and building off those data. So we have things in the work. It's a really slow process, though, and it's a frustratingly slow process. So I think as fast as we can. I think the tricky part is to get any of these. The patients are there for sure, who want these, right? It's just the money to get it's called the vector, and then to make the T cells, and you can only make so many. Even at Penn, where we do this all the time, we can maybe make one batch of T cells a month at best, right? And so if you try to do, you can't do too many trials, so you have to choose. But I hear you. I mean, we want to, and Kevin knows this, we try to push, but he knows it. We try to push the envelope as quickly as we can, but it's frustratingly slow. So I do think we'll get better ones out there, but in a way we had to do the first one first. But I hear I think again, I especially in the current landscape of research money, and I think you guys are the important force of, for letting people know how important this is, and in foundations like this one who actually give research money for right, and I think that's where all this happens, but it is slow.
Right. Thank you so much.
Welcome.
One more question? Go ahead.
Hi, thank you so much. My question is with the cartel therapy. I have a friend whose son was part of your trial, and he ultimately passed away in March. His initials were JD. I don't know if that means anything to you. He started the trial back in July, and he's been battling neuroblastoma. He was battling neuroblastoma for 7 years, and I know that he had 2 therapy sessions. So the first therapy session he had, they injected him with 1 million T cells, which I think is amazing. It's like one tiny syringe. They said it took 2 minutes, even though getting them all out is an all-day process. And then I know that he tried again. Are they the same T cells that you're injecting, or are you mixing them up a little bit? Because I know that the first transfer didn't work. I'm just curious.
Yeah.
I'm not trying to
No. I'm trying to
violate HIPAA.
No. That's fine. I'll give you a general answer. Yeah, it is amazing, right? You take all this blood out and it boils down to this syringe. But there's a lot of T cells in there. So the nice thing about CAR T cells, and this is where it's a living drug. So you can give a few, let's just say, and that turns into many as your T cells interact, whatever they're targeting, they proliferate and divide and then grow. That's not like a drug. Or you give a drug and then actually that drug gets metabolized and then goes away. This actually can live, and then it can live in like a reservoir for then if the tumor goes away, there's probably still T cells there that if it comes back, the hope is that they then grow back up and are able to fight the tumor. I do think in solid tumors, and part of the slides I tried to show you, there are all these forces that aren't amenable to T cells. So, thinking of this as a drug where you can give this, and we haven't gotten there, but and for brain tumors, we started doing this where you give it every 3 weeks because it doesn't last. I think is potentially a way to do this. Now, again, the expense of that is it's a lot. But my guess is he got the same dose, and some of that is again just clinical trial stuff because you can't keep going up at a dose until you prove one dose is safe. Would be my guess. But the hope is that if it did something, that if you give those T cells are probably gone, and then if you give more, they'll hopefully do something good again. So it's a good question.
Anyone else? No? Okay, thank you. Thank you, Dr. Bosse.